All right. Hello, everybody. Welcome to the Bazcast. We got a live webinar today. We are lucky to have some great people in the room with us. Thank you for joining. So we have Travis Duet, who is our soil health manager for Bass Hybrids. He specializes in biological soil management and research, focus heavily on functional soil biology, how we can leverage microbes to improve nutrient efficiency, root architecture, and crop resilience through soil microscopy, hybrid evaluation, targeted fertility strategies. His work is really to bridge the gap between the microscopic soil life and real world field performance. We also have Willie here, Willie Pretorius. He's the owner of Reconstruct-Ag. Willie has studied agriculture and plant breeding at the University of Pretoria, advanced degrees in pomology and plant physiology from the Stellenbosch University in South Africa, and has dedicated a large portion of his life to studying and promoting soil health. And we're lucky also to have Dr. Alok Adholeya joining us from New Delhi tonight on your end, founder and CEO of Umahari, where you guys are working to develop biologic-based solutions for sustainable North American agriculture, where you guys have integrated 40 years of research with nanotechnology innovations. Your background educationally is you have a master's in plant pathology, a PhD in microbiology, and spent time as a postdoctoral fellow with the USDA with a focus on mycorrhizal research. So I'd say we have some pretty smart fellows in the room here to have a conversation about mycorrhiza, what it can do for us in North American row crop agriculture, and not just that, but overall in plant health. And kind of the focus of our conversation today is going to be, can corn learn to feed itself, make itself more efficient? And so I appreciate all you guys joining me today here for this discussion. Dr. Alok, can you just, I guess, add on to that introduction and tell us what you're all about?
Well, thank you very much. And what you've said, I will just add a few words about me. With mycorrhiza, we have developed the technology of producing at commercial scale, which was a big deal. We used a very specific and unique technology to combine growing roots without shoot and then growing those mycorrhizal fungi on those roots under semi-synthetic media. So that's the turning point to bring mycorrhiza into commercial application situation. And then we continue working on those bugs to develop and understand about them much more and develop more and more species to create a highly broad spectrum product. And that's how currently we are promoting multiple species in one product of mycorrhizal fungi, which are functionally different in terms of providing response to different cropping systems.
Sure.
And that's what creates a unique and a very efficient product. Besides this, we have been working on bacteria and now for the nanonutrients to combine and making an impact so that we can build and we can win confidence among the farmers and the industries who can bring this product to market with confidence.
Okay.
Thank you.
Awesome. Well, thank you. I really appreciate you being here with us. Willie, why don't you say
Yeah,
a few words here for us?
Andrew, thank you, and Dr. Adholeya. It is a real privilege to have this opportunity to have the conversation with you guys today. The subject we're discussing today is something that I believe is becoming increasingly important to the future of agriculture. For many years, we have approached crop nutrition primarily from the perspective of what nutrients we need to apply to the soil in order to feed the crop. But plants evolve very differently. They evolve in association with an extraordinary complex of biological communities in the soil, of which mycorrhizae, bacteria, and other microorganisms effectively extend the nutrient acquisition capability of the root systems and help plants access nutrients that they may not otherwise struggle to obtain. So perhaps the question we should increasingly be asking is not simply how much fertilizer does a crop need, but rather how effectively can this plant access the nutrients that are already present in the soil. And I do a lot of soil testing. And when I look, that's one of the important things that I do, is to look at the total nutrient digest analysis. In other words, what is the total nutrients that's available and non-available? If you look at a soil and you look at the uh, definition of a soil, it's 25% water, 25% oxygen, and 45% mineral nutrients. And those mineral nutrients are, there's literally all the nutrients that's in the table is in the soil. And so all the mineral nutrients are there, but they are not available. They are not available. So that is the question of how do we get the plant to naturally access those nutrients like it happens in nature. This distinction has become important in the work I do through my company Reconstruct-Ag. When we evaluate soils, we frequently find substantial reserves of phosphorus, potassium, and other mineral nutrients. At the same time, we can find relatively poor biological activity or weak mycorrhizal populations. In other words, the nutrients may be there, but the biological mechanisms required to efficiently access and cycle those nutrients may not be functioning to their potential. And this has, in my view, been brought about by modern agriculture and the breeding technology that is losing these acquisition abilities.
So Willie, let me ask you this: with that, like, how do we go about, outside of, how do we grow that mycorrhizal population in the soil?
Well, first of all, you have to get a plant to associate with because mycorrhiza cannot live without an host plant association. That's how it grows and multiplies. If it does not have that association, it can't be fed, because it can only be fed through mutualistic association, where the plant provides the photosynthate to feed the mycorrhiza, and then it will grow and extend and become an extension of the plant's roots, which is like 10 and 100-fold. There's literally 100 miles, hundreds of miles of additional root systems that penetrate the soil in the areas where roots just cannot find the nutrients.
Okay.
And in that situation, that is how nature works out in the woods, in the
Sure.
natural prairie grasslands. That's how nature acquires the mineral nutrients.
Let me ask this question to you, Dr. Alok: is what makes a plant have that association or not? Because we know that there are some plants out there that will, and there's some that don't. What is that differentiation in the different cultivars?
So there are two aspects to it. One is that some of the plant families do not have mycorrhizal association, but there are only very few, only 3 family plant group, which do not make mycorrhizal association. Primarily, they are not making association because of their physiology and the sulfur which is involved in the process of metabolism. But on the other hand, the plants which requires support for nutritional raw material, they are looking for this kind of association. And the plants have a very unique situation where they are able to recognize which is a pathogenic organism and which is a friend. So once they recognize, they suspend their defense mechanism for milliseconds, and then the mycorrhizal fungi as a friend enters into the root system. So that's how the whole phenomenon works, and it's a need-based mutualistic or symbiotic relationship.
Sure.
Mycorrhizal fungi provides huge amount of surplus minerals in the available form to plants. And then in return, they get photosynthate the carbon for their own survival and multiplication.
Now, in what you've seen, is it, excuse me, does it make it more of like an on-demand need fulfillment system between that plant and the biology to then make things more efficient?
Absolutely. The more the deficient nutrient availability is deficient, the better is the symbiotic or better functioning symbiosis happens. So that's clearly understood. And the plant exchange or talking with mycorrhizal fungi is pretty clear. There are signaling molecules which allows plants to invite mycorrhizal fungi. The signaling molecules comes from the transporter mechanisms that the mycorrhiza provides. And that's how the whole association takes place.
So if we're looking at some of the needs of a corn plant, it's going to need NPK, it's going to need moly, zinc, cobalt, all these different things. Is there anything that the mycorrhiza cannot help provide in the mix of some of the macros or micros that the plant's going to need?
Not really, because the mycorrhizal fungi is non-selective to translocate nutrients. What they do is they release solubilizing enzyme in soils, and which makes the minerals get unlocked. Those are taken up by hyphae, and they are non-selective. So the supply system is of whole range of macronutrients to micronutrients. Those are coming into the system. In the current agronomic situations, when you apply high amount of phosphorus, the zinc mobility gets severely affected. Whereas the mycorrhizal fungi make sure that the zinc is sufficiently available to plants. So there is a non-selective absorption, but selective use by plants during this symbiosis.
If I can just jump in there, Andrew, when plants associate with mycorrhizae, they send signals to the mycorrhizal communities saying that at this moment in my physiological development, I need molybdenum. And that signal will be transferred to the microbiology, and they will find the molybdenum that's required and translated back to the plant where it's needed in its physiological state. And so it's with every nutrient that the crop or that the plant requires. The system, the signaling systems, there are like trillions of signaling systems happening all the time between the crop and the microbial community. And it's not only mycorrhiza that's involved. The other important is the rhizophagy cycle, which works in association with the mycorrhizal system. So it's an incredibly interesting phenomenon. How a plant says to his community which he's feeding, and that's where the mutualistic relationship comes from. I now need some nitrogen. I need phosphate. I need zinc. I need cobalt. I need molybdenum, and because all the different biochemical reactions that takes place in a plant need different cofactors, and a lot of these cofactors are minute amounts, which you find deficient in situations where these microbial communities are not present, and plants can grow. A lot of the plants, and as the big ag has developed agriculture, they've kind of overlooked all of those issues. They're only looking at yield, and all those factors you start losing them, and a lot of the components, your phytochemicals, which are part of your all your anti-cancers, all your anti uh, um, products that's required for good health is not being produced in crops that does not have a proper association with the root microbial communities, because the cofactors are not being there to provide the enzymatic reactions to produce these important components.
Okay. So if we have better nutrient acquisition that's happening through good mycorrhizal colonization, rhizophagy that's happening, a million billion little factories under the ground that are, I mean, rhizophagy, under a microscope is like a bloodbath that's happening under the soil. But one of the main things, if we get a system that's operating fully and really in, a fine condition, we're going to absolutely reduce our need for fertilizer. But if we're running a nutrient density test or seeing what's in the soil from different tests that's out there, we're not eliminating the need for certain things. We're just making things more efficient to then find out what we actually need to apply. Correct?
Exactly, exactly. That's why I regard the total nutrient digest analysis such an important issue. Most of your soils in the Midwest have ample of most of these nutrients, but when you move down to a place like Florida, that is a different kettle of fish. Your potassiums are low, and it's low in almost everything. So you have to kind of um, reintroduce those elements in a way that will not affect your microbial communities, and that becomes quite an issue.
So, I speak to a lot of farmers. Travis does as well. You guys as well. But when, there are people that are out there that'll say, okay, well, I've tried this and it didn't work, or I tried that and it didn't work. So what can we do, or what situations need to happen where we can improve upon the efficiency and effectiveness of a product like a mycorrhizal inoculant or different forms of biology that we can supplement what's out there and give it a shot in the arm? Like, how do we make this better so that we reduce the failure rate on some of these products?
I think Dr. Alok will probably be in a better position to answer that one.
Okay.
Basically, when we are talking about, I completely agree, first of all, that many a times farmers, definitely the growers, do see that they didn't find the response from the product. Now, the product as such has to undergo lots of test, validation, and confirmation that the product will function in different conditions and different situations.
Sure.
If that is not done, then definitely this kind of response will be seen. The most important and the key factor is that the microbes we are applying first should feel home.
Yeah.
And if they are feeling home, then they will function. And for them to feel home, we need to provide them all that they require to function in our product formulation. And that's how the product journey goes. So I would say if the product formulation and product application system is well understood, then the product will function. If we are able to understand the natural biogeochemical cycles in the rhizosphere, in the rhizoplane, then we are definitely able to develop a right kind of formulation.
Yeah, we from things that I've seen is, I mean, you get the system operating, it'll self-regulate a whole lot. You could have, like, in that root zone, the pH will be different, wildly different than what it is just a couple inches away, and even more so maybe in the middle of a 30-inch corn row. So we need carbon. They need to feel home, but they also need to be fed. So, some of the things that we've seen in the work that we're doing, and some of it is intercropping or interseeding in our corn and creating more root exudates that are happening in the entire system, feeding that into the profile. We have some situations, and Travis can speak to this a little bit differently, but hybrid to hybrid, you'll see different responses, and yield gain. Some don't like it. Most do if we have the system set up properly, in terms of what their response is. I mean, we have some that are, over a 30 bushel gain in reduced nitrogen and in a cover crop system or an interseed system. So, it leads into the questions, some of the work that we're doing on breeding and plant development. How much do you guys think that the genetic factor of it with the cash crop plays into its success in a more biologically friendly, or I guess better, quote unquote, better system?
Yeah, I have also done lots of studies with wheat and corn to understand why and what's happening that some of the modern varieties are not really mycorrhizal friendly or biological friendly. And the realization has been, and I think Travis might agree, and you would agree, that the the selection process of newer varieties and lines have missed out the below ground functioning of those plant species. And therefore, the behavior functions are different. Therefore, we need to go back to land races, bring those characters back into the new hybrids and or the GMO crops, and make sure that they are below ground biological friendly cultivars. Then the efficiency of the products that we supply would definitely be improved in those particular cultivars.
I see it very, very often that, I find a client with incredible soil health. I mean, the soil health is so good. And then when you look at the roots, there is the colonization percentages are very, very poor. We see this all the time. So, my endeavor and objective is to get a situation and to provide solutions that will marry these two, so that we will have the genetics that will work in a good soil health situation, because you have to have both. The one doesn't work without the other.
Now, Travis,
Yeah.
on our, yeah, so go ahead. Perfect.
I think that oftentimes when we look at big ag, when we're doing research and production, we're looking how the plant responds to chemical. What happens when I put on 200 pounds of nitrogen? What happens when I put a fungicide on this plant? We're not taking into consideration how these plants perform in a reduced fertilizer environment or under particular types of environmental stress. So I think a lot of that just comes down to how we look at these plants and what we consider to be a good plant for going out there to the field. And then it also goes into production as well. How are we producing these seeds that are then going to the farmer, and having the right biology on that seed is very important to also make these mycorrhizal relationships.
Precisely. The seed-based delivery is a very efficient way of delivering microorganisms, but also require very specialized understanding how they are coated on seed, how they are not affected by the chemistry on the seed, and remain persistently viable with the chemistry on seed during storage. So that's a very important aspect, and this is something which Willie and I have worked together to develop this understanding and help our clients to use the seed coating formulations for good response.
Yeah. Alok, can you give us your opinion on a mycorrhizal seed treatment versus an in-row treatment? Because I have my I just believe that seed treatment is way better than in growth.
Definitely, because we have to understand the close proximity and the initial head start for symbiosis, the availability of mycorrhizal spores, which are freed in soil and available to the newly emerging feeder roots, is the definitely a very efficient way. In furrow application, you need higher quantity of mycorrhizal spores to be applied in order to meet the demand of early mycorrhizal symbiosis.
We're picking the
And
placement right where we need it to start with. So I mean, it's got to be more effective and efficient, right?
that's right.
Yeah. Oh, and I can say, when we're looking at different systems that are out there in walking fields, especially early on in the season, where you'll see some of the differences maybe more than later on. But I also feel that later on in the season, if we have a really well uh, managed program to keep this alive, that we're going to finish really strong. We could delay senescence a little bit in the plant to then pack on more weight onto the grain and keep that system going. Then it makes everything. It increases nutrient density. It makes everything more efficient. Okay, so here's the one question I have. Some people ask it. One person in the chat here asked the question is, if we're in a more conventional system, but we want to start including some of these things into it, what's worse, a fungicide application or tillage?
Well, the tillage is more worse than the fungicide application, because fungicide application we can create formulations to deal with, but the tillage definitely reduces the population. And tillage is one of the major reason of reduced population, because of which we need to introduce functionally efficient mycorrhizal or microbial load into soil.
But if we are in some sort of a tillage system, that I mean the mycorrhizal network will work itself back together, correct?
Yeah, so that's the reason why the application of product is necessary.
Help bridge the
Because
gap a
the
little bit.
tilled soil, we need to bring efficient microbial symbiosis with plants. And if we provide threshold numbers next to the seed, then you are at least helping the current crops for its highest benefit that we can draw from this product or these product as based associations.
So
that's
in your work and what you're formulating with some of that you're creating the product with the spores in it that will be more durable against a fungicide application.
So one of the very important aspect in the application related and what you're saying is, we need to understand about mycorrhizal fungi. If they are making chlamydospores, chlamydospore is the stage on which they can sustain minus 10 degrees Celsius to 60 degrees Celsius. This means if the mycorrhizal spore and the product is in the right state, then every spore will germinate because they have completed the maturation cycle, and then they will germinate and colonize the plants. And then they can sustain the external conditions very easily to remain alive. So we need to understand that the right kind of product formulation with the right stage of mycorrhizal fungi will make a big difference.
Oh, that's pretty cool. Now, with that being said, what are like the next steps? What's the next part of the development, and like what's coming next with products that are like this?
Yeah, so more and more understanding, more and more mimicking the natural conditions so that the formulation becomes efficiently effective. If an early stage association takes place, the farmer is not worried. We need to understand the grower if he sees his crop is showing not the desired characteristics in the early phase, he gets very much worried. And we need to make sure that our product is on the rescue in the given situation. It's functioning, it's providing those advantages of enough nutrient supply so that the crop is not showing any stress symptoms. And that's the key from the formulation point of view.
All right. So in some of the work that we're doing on our discovery farm, research farm is, with the interseed and seeing how that is, how the plants respond to it, multi-species interseeding in between the rows.
That's a brilliant way, because if you're providing every inch of soil having active roots, the interconnection between plant through the mycorrhizal fungi is a very important aspect. I must mention that the mycorrhizal fungi is, when they are in association with plant, they are also supplying nutrients from one plant to another for creating balance.
So yeah, in that, with that translocation of some of these nutrients from one plant to another, are there things that we can put, or different species of plants that will help us increase some of that, then also have it in a program where it works in a corn system, where it's not going to, mess with the combine or add too much competition to the system? For most of the time, what we see is that if the system is moving properly, there's not a whole lot of competition that's happening. Maybe from some early season moisture for germination, but outside of that, it really doesn't exist from what we've seen. Are there certain things that we can put in our mix that would increase the likelihood of this, that quorum sensing from happening?
So, like you're talking about cover crop seeds, there are cover crop seeds combination which has legumes, pulses, and cereals combinations, because then, and the C3 and C4 plants. So when you're making this combination, the association between the plant of mycorrhizal fungi and the other bacteria, those are also equally important for symbiosis related benefits to crops, makes a very good situation. So selecting crops which are leguminous, pulses, nodule forming in the combination mix is a good idea, brilliant idea. And that's why the cover crop seed mix is something which is really providing additional advantages.
Yeah.
And it should be done.
We'll see what happens from last year to this year, because last year we had a 9-way mix. Travis, what's the mix this year? 13?
I think it's 12 or 13.
Yeah. So, adding a bunch of different, different plant families and getting that. I think the most important part of it is just not just having plants that are going to be a couple inches below, but multiple layers of root depth that's going to help move some of that stuff, because we're going to be building that soil from the bottom. So the more that we have that root structure, we have that rhizophagy happening for all the way down at the bottom, where some of that stuff might be locked up, and make it more available. So, Willie, you can speak to this, as, when we're looking at how do we know what's working or not? What are some of the things that we can do to find out if what we have, outside of what the monitor is going to tell us at the end of the year, or what the plants look like throughout the entire season, at the end of the day, like what's on the monitor still matters, and what tests are showing. If, and maybe that first year or second year, we're not seeing a huge jump in terms of overall yield, but we're building a legacy effect on that farm. So, Willie, how do we go about measuring and testing what that looks like?
Yeah, well, the most simple way to do it is through microscopy, and there's two measures that I use. I use what the soil spore density. In other words, how many soil spores do I find in a gram of soil, and then the other one is that are your roots being colonized? In other words, a percentage colonization. And I've actually reverted back to microscopy. I've been doing a lot of work on the PLFA and the NLFA your PLFA, when you see what is reflected in the mycorrhizal component, it's not correct, because there's many other bacteria that have the same point where it comes off the gas chromatograph. In other words, it influences the result. The NLFA is a much better relationship, and there's a good correlation between the NLFA that you find in nanograms per gram versus the percentage colonization. The only thing is, as we progress in time, I just need more points to actually get a confirmation, or get a better confirmation on the relationship with that curve. So I've actually, to make sure that my results are accurate, I've reverted back to microscopy in terms of soil density analysis and to root colonization. And that is the way that you can see whether the system is working.
And what about like genomic testing?
When you look at the prairies in the US, they had, some of the soils had in excess of 1,000 spores per gram of all the different mycorrhizae. Many of the results that I'm getting at the moment, we battled to get 50.
So if we have those spores that are out there,
it just shows you what the deterioration has been over time, because of lack of being fed and lack of being of sporulating and growing and increasing their numbers. If you don't feed them, I mean, ultimately, as Alok was saying, when they in a clematospore form, they can, they form thick soil cell walls. But over time, you can only live so long without being, without being revived, for lack of a better way of explaining that.
So if we have a lot of spores that are out there, you're saying, okay, like the prairie land. Some people, I've listened to some people say, okay, well, ideally we'd like to get to a 1-to-1 fungal to bacteria ratio. Well, that doesn't really exist outside of maybe a jungle floor or a forest floor, or more fungal dominant. Prairie lands, prairie land pastures, maybe 4-to-1 is going to be pretty good. But the second we start doing things in there and turning it into row crop agriculture, we're gonna mess that number up a little bit, and then it's gonna be more bacterial dominant. So if we're in a situation with, like, in a row crop situation, a corn or a sugar beet or beans of some variety, what would be, like, say corn, for example, what would be like an elite number for fungal to bacteria ratio?
Ideally, I would, if I can see a 1-to-1, I'm happy.
But the best North American soils were at best a 4-to-1. So like I don't think we can get to a 1-to-1 in a row crop situation. I think it's impossible.
I don't think it's impossible. I think it can be done. In fact, I've seen it.
How do we get there?
Well, marrying the genetics with the soil, and making sure that your soil health communities are diverse, and that you have at least a 1-to-1 fungal to bacterial ratio, and that your genetics fits the soil in which the crop is planted. You have to fit those two to get back to how nature functions.
I got a question that popped up here from Dave. Hello, Dave. I appreciate you being here. Are there things that we can do to help feed the mycorrhiza or the biological system during the winter months in the northern colder climates? Are the decomposing roots to feed the system, or do we need C3 plants to host during those winter months?
You actually need a growing plant. So any cover crop that will survive over that period should be, should give the mycorrhiza some comfort in terms of being fed. Now the only problem is that if you only have one cover crop species, it's going to only feed a selective number or species numbers of mycorrhiza, because all crops don't always attract the same components of mycorrhiza, and that's why when you put cover crops in, you try and accommodate the largest diversity of the mycorrhizal components.
So if we're in a more northern climate where we can't necessarily get certain things established, what's our best methods of increasing the likelihood of success in this type of an operation?
Let me come in here.
Sure.
First thing is that we need to understand the mycorrhizal fungi do require a dormancy cycle to become active, because in nature, when the vegetation activity is in dormant situation, the organisms are also supposed to be in dormant situation, and that's what it makes them to have higher virulence when the active season comes. So this is very normal that mycorrhizal fungi will undergo a dormancy situation when there is no crop and only roots which are harboring them, whether these vesicles inside the spore or the extra radical spores. They will all go secondary wall thickening during this time, which is what is required, and then they will become ready for the active season with higher virulence. So that's what is needed. Now, as Dave has asked about how we can improve and how we can help the mycorrhizal fungi or organisms to do better, my answer to that would be enhance the population during active time of those organisms so that they become in threshold number available to the growing crops in future.
Yeah. So then I guess my question to stem off of that would be is if we have cover crops that are out there in a wide variety, a lot of different numbers or species, and we have those different levels running through the soil surface and that are running deep, and we have stuff that is down there, our temperature is going to be a little bit better, but then they're going to be down there deeper for when we start to then jumpstart us better once we warm up, and then the living root gets back down there.
Absolutely. All stratas, if there are presence of microorganisms, that would certainly benefit the next active season.
All right. So one other question that came in here right now says, what are the physiological and biochemical changes that make successful mycorrhizal and plant signaling happen?
What is the mycorrhizal, the biochemical and
physiological?
Like
what are the changes that are going on that, hormones that help some of this stuff? What is, the question is, what exactly are the physiological and biochemical changes that make successful mycorrhizal and plant signaling?
Yes.
Like what's making that happen properly?
You see, it's a very interesting and complex system. The mycorrhizal fungi are not alone in the symbiosis process also. There are other organisms which are friendly with mycorrhizal fungi, and they are dependent on mycorrhizal fungi for carbon as well. So and these are the organisms which are doing signaling between plant and the mycorrhizal fungi. They are the one which are also facilitating several of the functions in the rhizoplane, including nitrogen fixation, including deaminase activity for protecting from pathogens. So it's an entire process through which the symbiosis has to be at their best in presence of the right kind of population of other organisms in the formulation.
Okay. Travis, when you're walking the discovery farm and looking out there and where we have some of these things that are happening, what are some of the differences that you see, boots on the ground, looking at it?
Generally, I'm seeing less disease pressure, thicker stems. I'm also seeing that there's a legacy effect. This is a building system. You might go out there and apply it, and it's just going to get better and better as you use regenerative practices. You're using cover crops. You're putting the right cash crops out there that are going to respond to mycorrhiza. So I think it's a matter of understanding this tool and finding the right ways to support it so it's able to give the farmer a better return on investment. I think that's one of the big things for American farmers right now is with fertilizer prices going up. The American farmers trained to go out there and yield, try to get as much out of that land as possible. But that's kind of being turned on its head now. So as we look at mycorrhiza, it's a really good way of kind of breaking us from that cycle.
Absolutely.
So if we have a system that is capable of housing AMF and we're populating properly, what are the impacts of then going out on top of that and putting out, just because, I'm doing this, and this at this time of putting out too much, fertilizer on top of it? Will it decrease the effectiveness, or are we just wasting money at that point and putting some of that stuff out there?
Well, I think Willie and I have had this conversation before, but we've talked about mycorrhiza and phosphorus. And that was a very eye-opening conversation because I think a lot of times when we're talking about utilizing this tool, we don't talk about that enough. So there are, and Willie probably can give us a better numbers on this, but there is a point when the phosphorus is going to actually negatively impact our mycorrhizal growth. So if you start thinking about these seed treatments that we're using to get the mycorrhiza out there, what are we going to see if we put like an in-furrow starter fertilizer?
I mean, ideally, is if we have the system working, we could eliminate a starter fertilizer, work on creating that better home, Alok, right, for it, feeding it properly. So having good biology, feeding it, keeping it safe, and then it's going to do a lot of the work for us for free. Oh, so I got a question for you, Alok. Is okay during your time, when you through what you were doing, and then your fellowship. What was the moment that you realized that the mycorrhiza is not just an interesting topic of research, but that it could really be something that could fundamentally change how we look at agriculture?
Yeah, the beginning of my getting interested into mycorrhizal fungi was from the fact that the several tree species cannot grow if they are not having mycorrhizal symbiosis. And that was the starting point where I got really interested to understand how the tiny organisms can make such a difference in plant system that they are dependent so much on them. But during my postdoctoral research and subsequently, and even before during my PhD program where I did my mycorrhiza and bacteria interactions, it was pretty clear that until and unless we understand the functionally relevant strains for a broader ecosystem and create balance of functional trait-based mycorrhizal species selection, we will not be able to really provide an effective product. And that was the journey that we took it further throughout, and we are continuing with it.
Oh, some people kind of look at that mycorrhizal network under the ground, kind of like the internet, everything's kind of connected. Oh, do you think that's a fairly accurate analogy, or does it kind of oversimplify what's going on underneath our feet?
Well, on the cellular level, if somebody sees it nuclear and the food is streaming within the hyphae, it looks like that you are on the plane and you are seeing on ground the cars moving on the road on both sides. So that kind of streaming that you see, which shows there is a life, there is a function, and there is a specific purpose of the mycorrhizal activity in plant system. And I have seen it because I have grown mycorrhizal fungi under in vitro environments. So I could see all stages of life cycle of mycorrhizal fungi. That was very interesting because that's how you understand how the function happens, what stage the mycorrhizal fungi will make spores, what stage they will move the energy to which direction. All those are very important.
No, my friend Baker here in O'Neill, Nebraska, asked, how much does it appear that we truly don't know, everything about mycorrhiza, in the sense that we know what it's capable of doing, but how much is it that we don't understand yet in this whole system?
With the changing agronomies, with the changing crop varieties, the science has to be at the pace to understand how the mycorrhizal fungi would still remain relevant on the top of the list of the buying list of the consumer. That's what we need to know and we need to learn every day. I think that's where we are.
Is it how the level of importance of what we're talking about today is, to me, it's of utmost importance. I don't think that it is viewed that way on a university level to where we should. So, if we're looking at different ag schools or different programs where people are learning to be agronomists, I think a lot of them don't even, these words never come out of their mouth in some of those programs. Like, how do we get some of these topics to be discussed in academia at schools that are producing the next, soil scientist, agronomist? What do we do to make it more relevant?
It's a full circle. When the biologics products are, which they are already now inroads, they are making a difference. The growers are receptive to using them, and it's growing. And it should grow if the more and more effective product comes to market. That would make the university and the research system to do more applied science and build up the new upcoming scientists, researchers, or technocrats for future, looking at more from the application point of view, which is you have rightly said. Like any other field, mycorrhizal research, we also have a huge amount of fundamental research, which is important at times for addressing some of the questions. You need that information. I have myself published 270 papers in journals, but the application science is definitely rewarding, and that reward system should reflect so that the focus in the university system also takes more preferences to applied research in the biologics area.
Oh, my friend Zero sent me a text message question saying, if all of the mycorrhizal fungi on the earth disappeared right now, what would happen?
Havoc. I would say only that.
That's a good answer.
It will be a catastrophe.
Let's see some of these. Yeah, I mean, I just know some of the systems that I run into that are maybe more conventional compared to other ones that are not. You can be standing in the middle of a road, looking on the west side of it and seeing a brilliant, beautiful field, and then to the right where there was all sorts of anhydrous tanks and tillage equipment and who knows what else that's going out there, and you just see it. It's one side of this road is alive and the other one isn't. So I would say
Yeah.
that in that little bit of a, of an example, there's probably not a whole lot of mycorrhizal activity going on the one side compared to the other. So it's brilliant when I see some of these people. I see it with my own eyes, or they send pictures across. I mean, it's pretty vivid, the difference. So yeah, havoc is probably a really good. That might not even be a harsh enough word, to be honest, in my opinion. Oh, yeah. Oh, if we're looking at, so obviously we're talking about like mycorrhizae and how we can get these plants to form associations and help feed themselves better, more efficiently. Outside of like the mycorrhiza, what other, what else do we need to be looking at? That's going to be, if we were to not put the spotlight right on AMF for a minute and put it on something else, what would be, in your opinion, and Willie, you can answer this also, and Travis, on the next thing that we, in line of, well, we need to focus on this, of course, but also we can't let this be a laggard.
I just think that the financial situation of the agricultural industry will drive this sector of the of agriculture forward. It's already becoming just too expensive to use fertilizers. And the other thing that worries me is we use the word yield too often as a as a aha, that's I get this yield. You should rather say I get this profit. If you can produce 150 bushels and make a decent profit, and I'd rather do that than produce 300 bushels and make less profit. And those numbers do become very obvious if you start removing your external fertilizer inputs.
Well, if we're creating a system in an environment on a field, that we're going to be making it more resilient. We're going to make it tougher against what Mother Nature can throw at us, where we can withstand an extended period of drought, or build aggregate in soils that's going to be able to hold more water. So all of those things, when it's bad, it's going to cost us money if we're not set up properly. And if some of this bad stuff does happen, like, I mean, there's a lot of areas right now that are, really suffering from drought. There are guys that I see that are out there that have built a lot of resilience into their system that are not suffering from that at the level that their neighbors are. Oh, I think it's super, super important. And then in the long run, building that resilience is then helping us save money. So we talk about it again, the legacy effect, year in and year out. We're just making everything better.
On one hand, the ROI is the key player in terms of the biologics, and I have no doubt in saying that the ROI with the biologics in place is going to be a driver. On the other hand, it's also very important that these products are functioning and giving the advantages, and we are providing the consumers the right kind of products which are together. Now, we need to also understand in the regenerative agriculture or a natural ecosystem, there are other organisms which are also playing a role with mycorrhizal fungi to plants. And we need to understand that all those players in the rhizosphere or rhizoplane, we need to create a right kind of blend of those so that the restoration of below ground activity for effective ROI is achieved.
Good answer. Thank you. I got one question here that says, all right, so for much of the last century, agriculture is focused on controlling nature, chemicals, genetics, and technology to make our ecosystems more predictable. It's never predictable. Your career has focused on working with nature instead. Do you believe the next agricultural revolution will require us to stop thinking like engineers and start thinking more like ecologists? And if so, what will be the hardest mindset for scientists, policymakers, and industry to change?
The functional biology is very important from my point of view. And the better understanding on the functional biology towards the highest ROI that the growers get would make sure that the ecosystem is functioning much better, and the irritants are taken care in the growing, in the food production in an effective way.
Yeah, I think a lot of it comes down to is people want better food, and more nutrient density, cleaner. You know, if we can remove some of these different products from systems, or at least reduce, you know, what some of the mass application is, remove nitrates out of the system, make things more efficient. It's going to be better for everyone. So, you know, that's of utmost importance. So, Willie, earlier you talked about microscope. Oh, sorry, go ahead, Doctor.
I was saying that, you know, one of the very startling example is that most of the food grain, cereals particularly, are deficient with, or they're providing less zinc because of the higher phosphatic fertilizer applied in field. And zinc is a very important component for humans. It brings the stress in humans, and it also have lots of other symptoms like I lost my hair. So this is something which is a reflection of the imbalanced nutrition that is coming to our cereal systems. So with the biological system coming back, these things are naturally addressed. We have seen over a period of 6 years in cropping sequences and systems that when we use threshold level of biologics, the food grain is nutritionally enriched and restored to the nutritional properties. That's also a very important function that we cannot ignore for a long time.
Well, absolutely. I could not agree more than with anything you said than that. Oh, earlier Willie was talking about, you know, when we're talking about some of the testing that we can do, and you've started to look a little bit more on the microscopy portion of it. Travis, right behind you, you have a pretty nice microscope that we used in the office there to look at certain things. So if we were looking at certain things, especially early on in germination, on what the association is going to be, what are you looking for when you're looking at that screen through the microscope?
So generally what we're doing is first looking from hybrid to hybrid. So the seed will be inoculated with a mycorrhizal seed treatment. It'll go into a germinator, and I'm generally taking samples from that at about 4 days, 8 days, 10, 12 days. And I'm looking at how colonization is going. So it's great for me to be able to quickly turn through a lot of hybrids and see. Sometimes we go to inoculate and we're not going to see hardly any. And then sometimes it lights up like a Christmas tree. So we're basically using a blue LED light where we're able to look at the mycorrhiza as it grows through the cells. It glows a bright green. And then next, I've really started taking a look at lot to lot. So looking at different production fields, looking at, you know, that seed was grown in a field. Was there a fungicide used? Was there not? Was there a bacterial-based product put out there? Things like that actually do change the way that these seeds are performing with added biology to them.
Yeah, I know there are, you know, some of the genetic differences that are out there, hybrid to hybrid. The now from Alok, on your end, if you're looking at how we're doing work in terms of our breeding, what would be your guess or thoughts or recommendations on how, when we're going through this process, to increase the likelihood of what we're producing and we're creating to have a better, like, what is our Rosetta Stone? What can we do to make our stuff better when it gets into an environment that will help it thrive?
You see, one of the very important factor for mycorrhizal symbiosis is the root architecture. and the root architecture and root surface. The higher the root hairs, lesser the mycorrhizal symbiosis needed. The lower the root hairs, the mycorrhizal symbiosis is much more needed because the plants will undergo stress fairly quickly. We should also not forget that as Willie mentioned, that huge hyphal network in soil due to active mycorrhizal spread provides 15 days, up to 15 days of water stress mitigation in plant system. So such things are very important to taken into account and provide a threshold activity in the soil area for crops to perform.
So once again, just helping build a more resilient system, right? So if we're building a more resilient system in seed production, in breeding, and all of these different things on what we're going on, because in our program, we're not running any fungicide, no insecticide. We're trying to throw as much stress at things as we possibly can, see what comes out. And then we just know that when it gets into the farm, it knows how to fight for its meal a little bit better than maybe something else.
It's brilliant that Travis have considered studying the different situation in the soils. For example, if there are other bioinoculant added and how they are impacting. These are some of the very brilliant way of tracking down the effectiveness of the biologics into the agriculture system.
Oh, in the work that you've done, the work that you're going to continue to do, what's next for you? What's the next step of the evolution? Umahari, why don't you give a little bit? What are you doing with Umahari?
So the Umahari, as I mentioned, that this is a company which is basically having never-ending appetite to take up the challenges and understand what is the problem that we can solve through our research core team and expertise that we have created. So that's how we do, and we have a pipeline research activity which will bring in solutions to various different problems. Specifically, in case of mycorrhizal fungi, we go to specific ecotypes and get the new strains, try to get them into production system, and then at the same time we study their functional mechanism. In this entire journey, we found some of the mycorrhizal strains are specifically highly efficient for oilseed crops. So we have taken those strains into the product. So this kind of learning is very important, which will keep on enriching for better and better and better product.
So on your website, and when we post this video online, I'll share a link to your website on there, and Willie's you as well. Is you talked about some of your areas of expertise, and one of them that stuck out to me was like environmental bioremediation. So some of the work that you're doing there, can you kind of talk about that a little bit? Why it's important? Because I think it's super important. There's a lot of stuff that's going out there that are, you hear in the news or people talking loud online about, PFAS and some of these other things that can happen. We can correct some of this stuff that's happening out there in the environment. So what are you guys doing? Or what do you think about that?
So I have been working, and my company is also working on some of the industrial overburdens. For example, fly ash. For example, phosphogypsum waste from the fertilizer industry. So these are huge areas which are covering ground, and they also pose environmental problems like particulate matter into air, clogging the drainage systems because they become airborne. So to help these kind of problems, which are man-made problems through the industrial activities, we select aggressive plant species which are highly microbial dependent, and we grow them in a shortest period of time with fastest ground cover. so that we can arrest these particles to become airborne, number one. Number two, we take some of the heavy metals not going into water bodies. So these are the organisms, immobilize them. So that's how we help industries to mitigate the impact of overburdens in several different areas. We have worked with the low saline desert land in Middle East to help them not to have the sand. Of course, we can't really help the entire desert to do that.
Sure.
But some of the urban, next to urban areas, we could create those kind of green patches through the effective microbial
So if
populations.
you're out there in a desert area, or you're building some of these green patches, an oasis, if you will, where we're kind of creating an environmental filter through that area that's going to help all of the surrounding ground around it. Well, I appreciate that work that you guys are doing there. Willie, so I have to give you the microphone here and talk a little bit about what you got going on, Reconstruct-Ag services you offer, and what you got going on. What's next for you? What does the future look like for Willie?
Well, my main objective is to help farmers who want to progress their regenerative agricultural situation, to marry the factors which we've discussed over and over on this podcast, to make sure that that genetics fits the soil biology, and measure all of that stuff and see how it stacks up, and how it would how I can emulate what happens in nature. That's really my main objective, and my business is helping them and providing some of the resources that I have at hand to incorporate into their systems to speed up the creations of of achieving that objective. So that's really in a nutshell what I do. And obviously there's an enormous amount of of intricacies um, that stems from trying to do that. And I'm finding different situations all the time, which makes it really interesting. But I'm involved in particularly two incredibly important projects at the moment, and the one is in Florida, where we are creating a regenerative ag system that will produce all the components that's required for food production, and it will all be sold on the farming operation. It is an exciting operation, and it's taking up a lot of my time.
Yeah, that's why you don't answer the phone, Willie, huh? You're down in Florida. But now the system that you guys are working on, doing what you got going down in Florida, do you believe this is something that you could use as example and then put this in other places fairly easily?
Yeah, absolutely. It will. I believe it will become a model for regenerative ag.
The one of the things I think about regularly is okay. Well, it's easy for me. I live in Nebraska. I can drive 20 minutes in any direction and get unbelievable protein, great vegetables that are I know the people that are growing all this stuff. I grow a lot of stuff myself. So then. But if we can have some of these that are in locations that will help reduce the size of some of these food deserts that are out there, I think is spectacular. Everyone talks about feeding the world, but sometimes we got to worry about feeding our neighbors first.
Yeah.
So, but if we can take that, that mindset and have it be taught across the globe, well, maybe that's how we feed the world, is by teaching other people how to feed themselves in a way. But, Travis, you got any questions?
No, I think we covered everything. I think this is a important conversation for farmers to be able to listen to understand this tool and how to use it and how to pair it with the right crop. So I'm hoping that everybody that tuned in today and listened to it has a better understanding walking away from this on how to use mycorrhiza.
Great.
Alok, you got
And
any close? Oh, sorry, Willie, go ahead.
If I can just say, I think it's when we get back to how the regenerative farming operation would fit into the community. I think it's vital for customers to know where their food comes from, and I'm seeing more and more of this happening, where the regenerative farming operation becomes part of the community, and the people know exactly where their food comes from. They know exactly what's used and how the farmer is endeavoring to reduce all these harmful stuff that is causing health and health havoc for us at the moment. And also in doing so, I think that, I will lay pounds to pennies that there's a direct relationship or a direct correlation between nutrient dense food and the use of chemical inputs. And also then the important thing is to get your crops to start producing these important phytochemicals, which can only be done if you have all the necessary micronutrients that drive those systems that synthesizes those products. And it also has a genetic line to it.
So I'll ask each of you this question: is if you had like a one genie wish left or a magic wand on something that you could do in the world of agriculture to make happen, what would that be?
Would one big ag to go away?
Big wish. Look, what about you? If you had a magic wand to make one thing happen in agriculture, what would it be?
I would say to all the viewers now and the future that the trust on biologics is necessity. The more we believe in them and more we create a space for them to be integrated into mainland agriculture to regenerative agriculture, I think the not only sustainability, profitability will also be on their side.
How about you, Travis? What's your wish?
I would say that more of a focus in agriculture on education. I think that a lot of farmers are being led around by, let's say, oftentimes agronomists with interests towards selling more products. When I sit down with farmers and we go over soil tests, we go over practices. A lot of them are blown away by being able to look at that data and start creating a farm plan that's not only going to give them a stellar crop, but also start healing the land. And I think that's where it's getting really exciting, is to see these farmers become very successful, while also increasing the amount of carbon that they're storing in their soil, increasing the amount of biodiversity. And then that scales all the way up to where you go onto these farms and you actually hear birds singing, you see insects flying. And so this is a profound impact, not only on our food system, but the world that we live in.
Well, gentlemen, I think we had a good conversation. I think we covered a lot of topics. If there were some questions that came up and I didn't get to it, I apologize. But I think that the time that we spent was impactful, and I think we're going to put this online so people can go back to it and review it. And once again, Willie, Alok, Travis, thank you guys so much for being here. And what are you pushing 9:30, almost 10 o'clock roughly there in New Delhi? Oh, I appreciate it. And it's a little bit goofy with the time change. So and then next time when you're back here in the States and back in Missouri, we'll have to connect and go have go have a meal or something. I appreciate it. Willie, Willie, we'll see you in about a week or so, up at our Discovery Farm Field Day. If you're paying attention to this, our field day is next Thursday, in Bird Island. We'll be going over all these, a lot of what we talked about today is, on display at our Discovery Farm, and a bunch of different things that we have going on there. Some that are focused on what we've been talking about, and other things that aren't, to show a difference. Really exciting, cool stuff to look at. And so thank you, everybody. I appreciate all the people that tuned in, that came in and out. So thank you all. And we will see you guys next time. Thank you.
Thank you.
Thank you.